Hello, this is your first lecture and we will be covering the scientific method. You will learn how to name the properties of life, list and explain the steps of the scientific method, describe the levels of biological organization, describe and use the scientific method, and distinguish between a hypothesis and a theory. So let's get started. Biology is the scientific study of life. When we study biology, we're looking at a really enormous diversity, and all life has got the following properties. There is order. This means that when you look at a living organization, there are predictable patterns. So, for example, if you look at the body of a human, you'll notice that we've got vertebrae that repeat themselves, these repeating units along our backbones. Regulation is also known as homeostasis. This is the ability to maintain your internal conditions. So it could be maintaining your temperature if you're a warm-blooded creature, but all creatures have to regulate the amount of food, amount of water, toxins, etc. in their body. All living organisms grow and develop from single cells, and so we'll be looking at some of the processes of growth and development later this semester. All living organisms use energy. Plants use solar energy to create chemical energy, and animals and other organisms use the chemical energy created by photosynthetic organisms. Living organisms respond to the environment. Just think about yourself in a hot Arizona day. You'll start to sweat, and that's your body's response to temperature change. All living organisms also reproduce, and when they reproduce, they pass on strands of their DNA to the next generation that inherit that. So there are rules to inheritance that are found in all living organisms. And last but not least, all living organisms evolve. They are subject to the processes of evolution, and we'll learn more about that later this semester. Depending on what sort of biology you study, you could call yourself a cellular biologist, a population biologist, or an ecosystem biologist. Life can be considered at many different scales. Those can range from a molecule all the way up to the entire planet. So from smallest to largest, we've got a cell. A cell is the smallest unit that can be considered alive and can reproduce itself. And a cell is made out of molecules. Molecular biologists will study molecules like DNA or protein and how they work within the cell. Cells join together to form tissues. A tissue is when you've got a bundle of cells that perform the same function. So you could think about your skin as being made up out of layers, and some of those layers have specific functions, like the epidermis, which is the outer layer of your skin, is there to protect your skin from pathogens. When you have two or more tissues that work together for a common function, we would call that an organ. So you've got lots of different organs in your own body, from your brain to your stomach to your skin to your reproductive organs, and each of those have got specific functions. Together, the organ systems of the body make up an organism. You are a single organism and can reproduce and are subject to evolutionary forces. Many organisms of the same species together are called a population. Populations of different species in the same area, if they interact, are called a community. So when I said you are an organism, I probably could say that I lied. You're probably better described as a community of organisms, because on your face you've got tiny little Demodex mites that live inside of your pores. In your digestive system, you've got all sorts of bacteria that live in your body and keep you healthy. This is part of the natural community that is you. When you've got multiple different communities in a single place, we generally refer to that as an ecosystem. Ecosystems incorporate both the living parts of the environment and the non-living parts of the environment. So we might be interested in cycles of nutrients or water when we study ecosystems. And at the highest level, we've got the biosphere. And as far as we know, there's only one of them, and that's planet Earth. And that's everything on the planet, both living and non-living. So throughout the semester, you're going to be basing your ideas on what I would call hypothesis-driven science. So it's a process of inquiry or asking questions that has a series of steps, and this is what makes biology a science, chemistry a science, physics a science, geology a science. We all use the same methodology of exploring information, and a key part of that is the hypothesis. A hypothesis is a tentative explanation for something that you have asked a question about. Another way of putting it, it's a proposed explanation for a set of observations. Now, what's really important about your hypothesis is that it has to be based in reality. It has to be based in science. So we come up with a hypothesis as an explanation, our best possible explanation for something that we observe. And then the next step would be to perform an experiment to see whether or not what we predict actually manifests. The kind of deductive reasoning that you find with the scientific method often takes the form of sentences that have an if-then statement within it. So if what I think is happening happens, then I'm going to have the following observations. We'll play with this idea more throughout the semester. Now, scientific method, a lot of books will have it described as a sort of a ladder of steps. You start out with observations. From those, you generate questions. The questions, you try to answer them with your best science possible, and you generate your hypotheses. Using your hypotheses, you design experiments to test your hypothesis. And you go ahead, do your experiments, and you try to explain what you find. Well, that's all very well and good, but the truth is that the scientific method is an iterative process. It repeats itself in a circular way, in that the observations from one experiment can become important for generating questions on the next experiment. So you don't just stop with your explanation. A new explanation can become an observation for another person's experimental process. Also, something that's really important about how science is studied is that your hypothesis has to be something that can be rejected. If you can't reject your hypothesis, then it doesn't count as a scientific hypothesis. You've always got to be able to say what you expect if you're incorrect versus what you expect to find if you are correct. So we're going to go through a little example. This is one of my favorites, and it's called the coral snake example. And my first question to you is, you've got two pictures of snakes. Which snake will you pick up? Now, most of my students will say, hell no, I'm not going to pick up any snakes. But the truth is, one of them will kill you and the other one will not. And so if you are brave enough to pick up a snake, you might choose one. And if you remember the poem from when you were a child, red on yellow, killer fellow, red on black, venom lack. I'm sure you have another version of it. That tells you that the snake on the top is the bad one, and the snake on the bottom is fine. So the question we have from this observation is, what advantage does a non-venomous snake have in possessing the color pattern of a venomous snake? Well, in nature, we have some repeated patterns. The first is called aposematic coloration. It's also known as warning coloration. And this is the kind of color pattern that most of us look at and think, wow, you're so pretty. Look at that frog, blue and black markings. It's a Dendrobates frog or a poison arrow frog. And in the wild, it eats ants, pulls toxins out of the ants' bodies, and then puts those toxins into its skin. It's warning any possible predator that it's probably not a good idea to try and eat him. Monarch butterflies, likewise, have got bright orange and black color patterns, and birds will not eat monarch butterflies because they once again have got poison in their body. All of those pretty little bugs and beetles at the bottom of the page have red and black markings. And in nature, if something's got red and black markings, it's an indication you probably shouldn't pick it up. It might bite you, sting you, poop on you, or otherwise cause you to feel upset. So maybe having striped patterns in those snakes is a form of aposematic coloration. Another option is cryptic coloration. This is a way to avoid being detected by others. Now, I've shown you in this picture some classic examples of cryptic coloration that are fairly innocuous. So we've got a kakapo parrot. This is a flightless parrot from New Zealand whose green feathers match very, very nicely with the fern forest that it lives in. We've got a little lizard who matches the granite rock almost perfectly, and then a butterfly which flutters around on the forest floor, and it looks very much like a leaf. Now, it's a hard stretch for your imagination to think about a pattern like a red and black striped snake as being cryptic. But in nature, you have to think about the context for the color pattern. And one of the examples I like to give in my class is that if I walked into class wearing full camo from head to toe, I would stand out. I would be pretty obvious. But if I was wearing dark green camouflage and I was walking around in a tropical forest, I would be very easily hidden. Likewise, if I came into class wearing a head-to-toe zebra suit, you would say I was definitely trying to get your attention. But if I was on the savannah and I was one of many in a herd of zebra, I would become almost invisible. You might not be able to tell where I began and the next zebra ended. So we have to think about the context of a color pattern in nature, not just what you see. Well, a guy called Ed made an amazing little experiment in Costa Rica. What he did is he got plasticine, which is basically like Play-Doh, and he made some snakes of different sorts. He made brown snakes and he made stripy snakes. Then what he did is he put the snakes either on a completely natural background or he put them on a solid background. He left those fake snakes out for 24 hours so that any potential predators could try and bite them. And being a dough kind of clay, anytime an animal tried to bite the snake, it would leave bite marks on it. So he could count the number of bite marks on each snake and get an idea of whether or not the snakes were attacked more or less than each other. So in this experiment, he has three hypotheses. The first hypothesis, H0, is also known as the null hypothesis. Null means no difference. This means that all the snakes would be attacked equally, no matter what kind of snake and no matter what kind of surface. The first alternate hypothesis is that the birds actively avoid tricolor snakes. This would be an aposematic coloration hypothesis. The second hypothesis is that the birds don't notice tricolored snakes, and we call this the camouflage coloration hypothesis. So what about the predictions? What do you think we might expect to find? Well, with a null hypothesis, you would find that no matter which snake or which background, the number of bird attacks would be the same. So if you made a bar chart, the bars would all be the same height. For the aposematic coloration hypothesis, what you would find is on a natural background, the tricolored snakes will be attacked less than brown snakes, and on a white background or a black background, the tricolour snakes will be attacked less than brown snakes. So no matter which background you're on, brown snakes get eaten more and stripy snakes get eaten less. Camouflage coloration hypothesis would be that on natural backgrounds, the tricolored snakes are attacked less. But on a solid background, the snakes would be attacked at the same rate. So what did his experiment find? Well, let's have a look at the actual results and think about it for a second. No matter which background you're on, they did not eat stripy snakes. So it's not the null hypothesis, not the camouflage hypothesis. But what we would say is that the aposematic coloration hypothesis was supported by the data. Now, using those terms is really a precise use of words. In biology, we don't say, I proved my hypothesis correct. We don't prove things in biology. You get data that support your idea or data that reject your idea. So be careful about how you use your vocabulary. Coral snake color patterns, at least in the place that Ed studied them, are aposematic and do not provide camouflage. I have another really cool article here for you with a video to watch. This is a little article that came out this spring on how parrots help each other to obtain food rewards. And I'd like you, if you have some time, to go into the PowerPoint, click it, and have a look at the experiment and how it works, so you can get a real live modern experiment as well. All right, I'm going to finish up with discussing theories versus hypotheses. Now, in biology, we have a bit of a problem. The problem is that in science, we have specific terminology that has specific definitions, but in common English, there are different definitions for those same words. And so you'll hear people saying things like, oh, my theory is that it's going to rain tomorrow. And all the biologists in the room should slap their foreheads and go, oh no, they said the wrong thing. What you meant to say was my hypothesis, my tentative guess. The theory is something a lot more substantial. In biology, we don't have laws, we have theories. And I have this mental image that I'd like to give you, and it's the multi-leg table. Imagine a table with many legs, and each leg is a hypothesis that has been tested, and that hypothesis has been supported by the data. Now, if you have just one leg for a table, the tabletop will not be stable, it will fall down. So you cannot have a theory, which is represented by the tabletop, with just one hypothesis. A theory has its strength in that many, many hypotheses on that same topic have continued to add support to those ideas. So in biology, we will be dealing with a whole bunch of different theories that you'll be able to learn more about. One of the important ones being the theory of evolution. Evolutionary theory is supported by the laws of physics, the laws of chemistry, and huge numbers of experiments, fossil evidence, molecular data. And what's really important about a theory is, like a hypothesis, it allows you to generate predictions and it allows you to do things. Modern medicine, for example, the genetic algorithms on your phone that allow your phone to run quickly, all of those are based in evolutionary theory. So it's a really powerful idea that when you have lots of small hypotheses, that they can sum up to something that's very stable and very strong, and that you can build more concepts on top of that. So be careful about the term theory. It's a broad, well-substantiated concept versus a hypothesis, which is a tentative answer to a question. It only takes one experiment to reject a hypothesis. But just because you reject a single hypothesis does not mean to say that you would reject an entire theory, especially if there's lots of support in other areas. So with our multi-legged table, If you took off a few legs here and there, the table would still be stable.